Following the establishment of prokaryotic life during the Archean Eon around 3.8 billion years ago, the second stage of life's evolution on Earth unfolded amid profound geochemical and geological transformations that reshaped the planet's habitability. Oxygen, in its free molecular form, O2, posed a lethal threat to the predominantly anaerobic microbes that dominated early ecosystems, as it could generate reactive oxygen species, ROS, like superoxide radicals that damaged cellular components such as proteins, lipids, and nucleic acids in reductive environments. This toxicity stemmed from oxygen's high electronegativity, which disrupted the electron transport chains in primitive metabolisms reliant on fermentation or chemosynthesis. Initially, photosynthetic organisms were anoxygenic, utilizing hydrogen sulfide or iron as electron donors without releasing O2, allowing them to harness sunlight while avoiding oxidative stress. However, evolutionary pressures, driven by nutrient scarcity and environmental gradients, favored adaptations that exploited water as an electron source, leading to the innovation of oxygenic photosynthesis. This breakthrough, pioneered by ancient cyanobacteria around 3.5 to 2.7 billion years ago, split water molecules to produce O2 as a byproduct, marking a pivotal shift that not only provided a more efficient energy pathway, but also began altering the global atmosphere. Cyanobacteria, with their ability to fix carbon and nitrogen while generating oxygen, proliferated in shallow aquatic settings, setting the stage for the Great Oxidation Event, GOE, approximately 2.4 billion years ago, when atmospheric O2 levels rose dramatically from trace amounts to about 1 to 10% of modern levels. As cyanobacteria thrived, their oxygen output triggered cascading environmental changes, particularly in the oceans, which were initially rich in dissolved ferrous iron, Fe2+, from volcanic and hydrothermal sources. This O2 reacted with the iron, oxidizing it to ferric forms, Fe3+, that precipitated as insoluble oxides like hematite and magnetite, forming vast layered deposits known as banded iron formations, BIFs. These striking red and black striped rocks, accumulating on ocean floors from as early as 3.8 billion years ago, with peaks between 2.8 and 2.4 billion years ago, served as geological sinks that sequestered oxygen and depleted oceanic iron over time. By around 2.5 billion years ago, as BIF deposition waned due to iron exhaustion, ocean salinity remained elevated, potentially 5 times higher than today, owing to ongoing volcanic inputs and limited continental weathering to dilute salts. Concurrently, Earth's interior was cooling, with solidified crustal slabs from the primordial era sinking into the lower mantle, while buoyant mantle plumes rose, instigating episodes of mantle overturn. These convective upheavals, occurring sporadically in the Archean and Paleoproterozoic, redistributed heat and materials, uplifting basaltic crust to form emergent landmasses and shallow marine shelves. Such environments, bathed in sunlight-penetrating clearer waters, provided ideal niches for cyanobacteria to flourish, accelerating oxygen production and further oxidizing the atmosphere. The once iron-green oceans transitioned to a familiar blue hue as soluble metals diminished, illustrating the coevolution of life and geosphere. Microbial activity not only detoxified waters but also stabilized climates through enhanced weathering and carbon burial, fostering conditions for more complex biospheres. This oxygenation represented a double-edged sword in life's journey toward complexity, acting as both a crisis and catalyst that propelled evolutionary innovations en route to civilization. While the GOE triggered a mass extinction among obligate anaerobes unable to tolerate rising O2, survivors adapted by developing antioxidant enzymes like superoxide dismutase and catalase, harnessing oxygen's energy potential through aerobic respiration. This metabolic upgrade unlocked vastly more ATP from glucose, up to 18 times that of anaerobic pathways, fueling diversification and ecological expansion. Yet the narrative of Earth's biosphere intertwined with cosmic events, as interactions with neighboring galaxies influenced stellar dynamics. Around 5 billion years ago, the Milky Way's encounter with the Sagittarius dwarf galaxy, a small satellite system still merging today, sparked intense star formation, bursts through gravitational perturbations, potentially contributing to the condensation of our solar nebula and the birth of the Sun and planets about 4.6 billion years ago. Supernovae from these newborn stars unleashed cosmic rays that could erode planetary atmospheres or mutate genomes, though their direct impact on early life remains speculative. Far later in the Neoproterozoic era, these galactic ripples may have indirectly echoed through enhanced cosmic ray fluxes, but the primary drivers of subsequent global changes lay closer to home, in tectonic and climatic feedbacks that continued to sculpt the stage for life's ascent. Transitioning to the third stage of life's evolution, Earth endured one of its most extreme climatic trials during the Cryogenian period around 720 to 635 million years ago, an episode dubbed Snowball Earth, where ice sheets engulfed the planet from poles to equator, plunging it into a frozen stasis. This global glaciation, comprising at least two major events, Sturtian approximately 720, 660 Ma and Marinoan approximately 650, 635 Ma, arose from a confluence of factors. The breakup of the supercontinent Rodinia positioned landmasses near the equator, enhancing silicate weathering that drew down atmospheric CO2, reduced greenhouse gases from diminished volcanic activity, and possibly orbital variations or cosmic influences like increased cloud nucleation from galactic cosmic rays. Though supernovae from ancient Milky Way Sagittarius interactions could have amplified cosmic ray bombardment, eroding the heliosphere and seeding clouds, the primary trigger was likely a runaway albedo feedback. Ice reflecting sunlight, cooling the planet further until oceans froze solid, with thicknesses up to kilometers. This cataclysm induced a mass extinction, smothering photosynthetic life under perpetual twilight and halting the carbon cycle. Yet pockets of survival persisted in refugia, beneath thin equatorial ice in hydrothermal vents, or subglacial lakes where geothermal heat and nutrient circulation sustained prokaryotes. These hardy microbes, shielded by Earth's dynamic systems, from mantle-driven tectonics to oceanic currents, highlighted life's embeddedness in larger cosmic and planetary frameworks, where the solar system's stability and galactic milieu indirectly modulated surface conditions. In the aftermath of Snowball Earth, as volcanic CO2 accumulated under the ice and erupted through fissures, triggering rapid deglaciation and a greenhouse rebound, surviving prokaryotes diversified, paving the way for eukaryotic emergence, though this transition predated the glaciations with roots tracing back to 1.5 billion years ago. Prokaryotes adapted through endosymbiosis where aerobic bacteria were engulfed by host cells but retained as mitochondria, providing efficient ATP via oxidative phosphorylation, while photosynthetic cyanobacteria became chloroplasts in plant lineages. This serial endosymbiotic theory explains organelles' prokaryotic traits, their own circular DNA, 70S ribosomes and double membranes from engulfment. A single eukaryotic cell could house thousands of mitochondria, amplifying energy output and enabling larger sizes. Eukaryotes grew up to 1 million times bigger than prokaryotes, with compartmentalized nuclei protecting elongated DNA from oxidative damage in O2-rich waters. Nuclear membranes, possibly derived from infolded plasma membranes, safeguarded genetic material, allowing complex gene regulation and multicellularity. This surge in complexity defied the second law of thermodynamics' tendency toward disorder, as life locally decreased entropy by organizing matter, fueled by solar energy, while increasing overall universal entropy through waste heat and byproducts. Post-snowball eukaryotic innovations spurred the Ediacaran and Cambrian radiations, transforming simple microbial mats into diverse ecosystems and underscoring life's resilient trajectory amid planetary upheavals. As the Proterozoic eon progressed, Earth's tectonic forces orchestrated a grand assembly of landmasses, culminating in the formation of the supercontinent Nuna, also known as Columbia, around 1.8 to 1.5 billion years ago. This colossal continent emerged from the collision and accretion of smaller cratons, driven by plate tectonics that welded ancient crustal blocks together along orogenic belts. Nuna's vast expanse provided expansive habitats for cyanobacteria, the pioneering oxygenic photosynthesizers that had already begun transforming the planet's atmosphere during the Great Oxidation Event earlier in the Paleoproterozoic. In the lakes, rivers, wetlands, and estuaries dotting this supercontinent, cyanobacteria thrived, harnessing sunlight to split water molecules and release free oxygen as a byproduct. However, much of this oxygen was initially consumed in oxidizing dissolved iron in the oceans or decomposing organic matter from dead cyanobacteria, limiting its accumulation in the atmosphere. On land, the story differed. Rapid burial of cyanobacterial remains under sediments prevented complete decomposition, allowing surplus oxygen to escape into the air. The sheer scale of Nuna amplified this process. Its extensive surface area accelerated weathering and erosion, delivering phosphorus and other nutrients to coastal zones, further fueling cyanobacterial blooms. As a result, atmospheric oxygen levels surged, climbing toward 10 to 20% of modern concentrations, a critical threshold that would later enable more complex life forms to emerge from the shadows of microbial dominance. Yet supercontinents are ephemeral in geological time, and Nuna's reign was no exception. By approximately 1.3 billion years ago, rifting and mantle upwellings began fracturing this landmass into smaller continents, dispersing them across the globe. Tectonic inexorability soon reassembled fragments into a new supercontinent, Rodinia, around 1.1 billion to 750 million years ago, straddling the equator and encompassing nearly all of Earth's continental crust. In Rodinia's equatorial regions, subduction zones where denser oceanic plates plunged beneath lighter continental ones played a pivotal role in mantle dynamics. Accumulated slabs of subducted material stalled in the mantle transition zone, a boundary layer at 410, 660 kilometers depth, before cascading downward in catastrophic slab avalanches toward the core mantle boundary. These descending slabs cooled the outer core, disrupting convective flows and potentially weakening Earth's magnetic field, shifting it from a stable dipole configuration to a more chaotic quadrupole state with multiple poles. This diminished geomagnetic shield left the planet more vulnerable to external cosmic influences, setting the stage for environmental upheavals. Meanwhile, Rodinia's thermal insulation over the mantle fostered superplumes, vast upwellings of hot rock that contributed to its eventual breakup around 750, 600 million years ago, fragmenting into proto-continents like Laurentia, Baltica, and Gondwana. Amid these terrestrial shifts, cosmic drama unfolded on a galactic scale, as the Milky Way continued its protracted interaction with the Sagittarius Dwarf Galaxy, a satellite system that has been merging with our galaxy over billions of years. This encounter, with major passages occurring around 5 billion years ago and more recent brushes, triggered starburst phases, intense episodes of star formation fueled by gravitational disruptions. Within millennia, many of these newborn stars exhausted their fuel and detonated as supernovae, unleashing torrents of cosmic rays, high-energy particles that traversed the interstellar medium. Earth's weakened quadrupole magnetic field during the late Neoproterozoic, around 600 million years ago, offered scant protection, allowing these rays to penetrate the atmosphere more readily. According to some models such as the nebula winter hypothesis, cosmic rays seeded cloud condensation nuclei, fostering thick global cloud cover that reflected sunlight and plunged the planet into severe glaciations, the cryogenian snowball Earth events, where ice sheets enveloped even equatorial regions. Alternating with periods of extreme heat from volcanic CO2 releases, these cold snaps dropped atmospheric oxygen to Archean-like lows, as photosynthesis stalled under perpetual ice. Mass extinctions ensued, decimating eukaryotic microbes and simple multicellular forms, yet these crises birthed opportunities. Survivors adapted, while cosmic ray-induced mutations and oxygen fluctuations accelerated genetic innovation, fostering novel body plans and ecological niches. As the starburst waned and supernova activity subsided, Earth's core dynamics stabilized, reverting the magnetic field to a robust dipole by the Ediacaran period, 541 million years ago. Resurgent photosynthesis by cyanobacteria and emerging algae replenished atmospheric oxygen, restoring it to pre-glaciation highs. Beneath the surface, Earth's gradual cooling introduced another transformative process, the leaking Earth phenomenon. In the planet's hotter youth, subducting oceanic plates released water bound in hydrous minerals back to the surface via volcanism, maintaining stable sea levels. But as mantle temperatures dipped below approximately 650 degrees Celsius around 600 million years ago, these minerals retained water, dragging it deeper into the upper mantle and beyond. This inexorable subduction cycle sequestered about 3% of Earth's seawater, equivalent to an ocean's worth, into the interior over tens of millions of years, causing global sea levels to plummet by up to 600 meters. Exposed continental shelves expanded dramatically, creating sunlit shallow marine environments teeming with nutrients eroded from inland rivers. Enhanced weathering on these burgeoning landmasses further boosted oxygen production as silicate rocks reacted with CO2 to form carbonates, drawing down greenhouse gases while liberating ions that fueled algal blooms. These nutrient-rich shelves became evolutionary hotbeds, where fluctuating oxygen levels and post-glacial stresses, combined with genetic mutations from lingering cosmic rays, spurred rapid diversification. This confluence of geological, atmospheric, and cosmic factors ignited the Cambrian explosion around 541 million years ago, a biological renaissance where animal phyla burst forth in unprecedented diversity. In mere tens of millions of years, a geological blink, complex ecosystems arose, featuring arthropods, chordates, and mollusks with hard parts, eyes, and predatory adaptations. The expanded continental shelves, bathed in sunlight and enriched with nutrients, provided vast arenas for experimentation, while oxygen spikes enabled larger bodies and active metabolisms. Genetic mutations, amplified by environmental stresses and possibly cosmic radiation, unlocked developmental toolkits like Hox's genes, allowing modular body plans to evolve rapidly. Mass extinctions had cleared ecological space, and rising calcium in seawater from enhanced weathering facilitated biomineralization, armoring creatures in shells and exoskeletons. Though debates persist, whether oxygen was the prime trigger or merely a permissive factor, this era marked life's leap from simplicity to sophistication, a testament to Earth's dynamic interplay of fire, ice, and stardust forging the foundations of modern biodiversity.